Hello everyone, welcome back to another session of respiration in plants. I am Raji here, botany, a lecturer from NextGen Academy. So till now what we have discussed that how the respiration proceeds in plants, right? So we had a discussion on the basic introduction of respiration. We had dealt with cellular respiration. We had a discussion on what are the different respiratory substrates that can be involved in the process of cellular respiration. And then we have started with the aerobic respiration concept, right? And before that, we also had a discussion on what are the different types of respiration, mainly aerobic as well as anaerobic. And I also told you what are the different steps that are present in aerobic respiration. So as I'm repeating again, so it will be easy for us to proceed for today's class. Aerobic respiration, as we all know, it happens in the presence of oxygen. That means breakdown of complex compound in simpler form in the presence of oxygen and by releasing energy, and this energy is used to form ATP, right? So aerobic respiration has been a very important event in all aerobically respiring organism, because it is the event that generates energy, right, and which helps in doing all the functions of the organism. So in plant also, since we know that plant is aerobic, so the type of respiration that plant also undergoes is aerobic respiration. So in the previous class we have discussed the common pathway for both aerobic and anaerobic respiration, that is glycolysis, which is also known as EMP pathway due to the name of the derived from the name of the scientist, that is Embeden, Meyerhoff and Parnas pathway, right? So when we talk about glycolysis, whether it is aerobic or anaerobic respiration, glycolysis will be the first step, right? And what happened in glycolysis? The substrate, what was the substrate for glycolysis? Sugar, that is glucose, is partially broken down into pyruvic acid. So as we discussed, glucose is made up of 6 carbon atom. It is broken down into 3 carbon atom compound like pyruvic acid or pyruvate, right? So now since the product contains only 3 carbon atoms, so the number of product molecule will be twice. So one glucose molecule is partially broken down into 2 molecules of pyruvic acid, right? And when we talk about the site of the reaction, it occurs in cytoplasm, whether it is a prokaryote or eukaryote. That means in bacteria, in fungi, plant, animalia, everywhere, protista, it will occur in the cytoplasm of the organism. So that is about glycolysis. So now after glycolysis, once the pyruvic acid is formed, right? So the fate of pyruvic acid depends on the type of organism, right? So if the organism is aerobic organism, or if it's an aerobe, or if it is an aerobically respiring organism, pyruvic acid will straight away undergo the next step, that is called as link reaction, right? On the contrary, if the organism is an aerobic organism, so instead of undergoing link reaction, which is a part of aerobic respiration, if the organism is anaerobic organism, it will not undergo link reaction. So this pyruvic acid will directly undergo the process of fermentation or anaerobic respiration. So that means the similarity between aerobic and anaerobic respiration ends in glycolysis. That is the reason glycolysis is said to be the common step for both aerobic and anaerobic respiration, right? So today what we will do here, first, since in the previous session we had studied already, had a discussion on the formation of pyruvic acid. So today we will talk about fate of pyruvic acid, right? So as I was telling you, the fate of pyruvic acid is decided by the type of organism, right? Like for example, if the organism is aerobic, right? So aerobically, the pyruvic acid will be completely converted into carbon dioxide and water, right? At the same time, anaerobically, right? When we talk about anaerobically, it will undergo the process of anaerobic respiration, which can also be defined as fermentation, right? Now, when we talk about fermentation, two types of product, right? Fermentation is of different type, but when we are within the textbook, within our NCERT textbook, two types of fermentation has been listed in the textbook. One is ethanol fermentation, so the byproduct will be ethanol and carbon dioxide, right? And the other product here is lactic acid. So here fermentation or anaerobic respiration, two types is given in our textbook. One is ethanol, right, fermentation, and the other one is your lactic acid fermentation. So today we will discuss the fate of pyruvic acid, what are the different steps if it is undergoing aerobic respiration, and what are the different process or methods or the steps if it is undergoing anaerobic respiration, right? So first we will have a discussion on fermentation. Right. Now, when we talk about fermentation, it is an anaerobic process. Always remember that. So, fermentation is an anaerobic respiration. So, it is an anaerobic process. So, it doesn't involve the presence of oxygen, right? So, either it can happen in the complete absence of oxygen, or it can happen in a partial presence of oxygen, like your muscle cells or lactic acid bacteria, where your lactic acid is produced as a byproduct, right? So there are few things when we need to consider when we are talking about fermentation. So now fermentation, when we talk about, it is an extracellular process, right? And it is enzyme mediated. Right? So one of the first documented, discovery of fermentation is the discovery of alcohol production, right? So wherein sugar was converted into alcohol with the help of yeast, right? So the earliest documentation of fermentation was sugar converted into ethanol in the presence of enzyme yeast, right? Which also released gases and later was identified as carbon dioxide. Now the yeast, the enzyme that was responsible for this reaction was zymase, right? So this is the earliest concept that is available for us. So fermentation was discovered very much early. So now when we talk about fermentation, here see, I haven't started with pyruvic acid, although what we are studying is the fate of pyruvic acid. But if somebody asks you, if it is asked in the exam, what is the substrate for fermentation? The substrate for fermentation is not pyruvic acid. The substrate for fermentation is sugar. And which sugar are we talking here about? Glucose, right? But always when we talk about fermentation, glucose is not used as a substrate. Maybe the substrate is complex carbohydrate, like it can be starch mostly. So starch has to be first broken down into simpler one, and then the process of fermentation will start, right? So as we know that the basic step of both aerobic and anaerobic, the first primary step is said to be glycolysis. So here also when we talk about formation of alcohol, right? So if I want to talk about the formation of alcohol, so first we will talk about ethanol fermentation. Right? So in ethanol fermentation, so this is the first fermentation that is given in our textbook. Ethanol fermentation, so what will happen here? What is the substrate here? Sugar, right? Like for example, the type of sugar that is most commonly used by most of the organism is glucose, right? So sugar or glucose will first undergo partial breakdown to form pyruvic acid. Right? So we already studied sugar is made up of 6 carbon. Pyruvic acid is made up of 3 carbon molecules. So one molecule of sugar forms 2 molecules of pyruvic acid. Now this process is what we studied in the previous class as glycolysis, right? So although see here we are studying fermentation, glycolysis is an integral part of anaerobic respiration also, right? So pyruvic acid is formed. Now what is the fate of pyruvic acid? Since we know that the organism is anaerobic, pyruvic acid has to undergo anaerobic respiration. So what will happen to pyruvic acid here? Pyruvic acid first will be converted here directly, right? It will be converted to pyruvic acid will directly be converted to alcohol, right? So what is the alcohol here? Ethanol, right? So this is how we say here, right? So pyruvic acid is converted to ethanol, but there are few in between steps here that we need to remember, right? So this is how it is given in our textbook, but sometimes they ask the question in detail, right? So what kind of questions and what else we need to remember here? Let us see here, right? So now what we will see here, what we will break it down. So pyruvic acid, a 3-carbon compound, is first converted to acetaldehyde. Right? Now, what is acetaldehyde? Acetaldehyde is a 2-carbon compound. See, pyruvic acid is a 3-carbon compound. Acetaldehyde is a 2-carbon compound. So, there is 1 carbon less, right? So, that 1 carbon is released in the form of carbon dioxide, right? So, release of carbon dioxide, the process is called as decarboxylation, right? And decarboxylation is catalyzed by which enzyme? Decarboxylase, right? So the primary enzyme that is participating in the process of alcohol fermentation is decarboxylase, which converts pyruvic acid into acetaldehyde, right? Now later this acetaldehyde will finally be converted into ethanol, right? Now here when we talk about ethanol, right? So this process, acetaldehyde to ethanol, it is a reduction process, right? So what is the reducing agent here? NADH plus H plus, right? So this is the reducing agent which in the process gets oxidized. So reducing agent get oxidized, but acetaldehyde gets reduce. So this is the reduction process. So I have seen a question, what except the electrons or hydrogen during alcohol fermentation? So it is acetaldehyde which is not included in the book, in the textbook. It is directly pyruvic acid from ethanol. So what collect or what takes the hydrogen atom or electron from NADH? It is taken by acetaldehyde. So acetaldehyde is getting reduced to ethanol, whereas NADH, which is the reducing agent, is getting oxidized to NAD plus, right? Now here the enzyme that is participating is alcohol dehydrogenase, right? So you can see here 2 enzyme is participating in the process. One is decarboxylase, the other enzyme is alcohol dehydrogenase. In the textbook, they have given both, together and the ultimate product as ethanol. But it is very important for us to remember acetaldehyde is an intermediate product during the formation of alcohol, right? So during alcohol fermentation, what are the byproducts? You have ethanol as a byproduct. You have carbon dioxide as a byproduct. Remember, 2 pyruvic acid means 2 carbon dioxide, 2 molecules of acetaldehyde. So you have to remember that thing, right? So this is our process of alcohol fermentation. So now when we look at the process, these are the byproduct, right? So energetically, right? So when we talk about how much energy is released here, right? So you cannot see any kind of energy or ATP getting released from pyruvic acid to ethanol. You cannot see any kind of NAD plus NADH plus being released or any kind of ATP being released. See, one very important thing, many times students, they don't remember we are using NADH here. What they recall, we are using NAD and NADH is produced. NADH is not produced here. NADH is used here. So no energy equivalent, that is neither NADH2 or ATP is formed during the process from pyruvic acid to ethanol. So only source of ATP generation during the process of fermentation is from glycolysis. So from glycolysis, how many ATP yesterday or the previous class we have discussed? Only 2 ATP. So energy released during fermentation, right, is only 2 ATPs, right, and that 2 ATP is also coming through which process? Through glycolysis, right? So in anaerobically respiring organism, how many ATPs are produced? Only 2 ATPs are produced. And what is the source of that 2 ATP? The source of that 2 ATP is directly from glycolysis. That is the reason when we talk about the difference between ATP in aerobic and anaerobic respiration, it is huge. It is 19 times more. Here it is only 2 ATP. There it is how many? 38 ATP. So that there is a huge difference. So now why it is a huge difference here? Why there is a difference in energy production for aerobic and anaerobic respiration? Because when we talk about anaerobic respiration, the product, what is the product here? Ethanol. It is a partially breakdown product. It is a partial breakdown product, right? So partial breakdown product of glycolysis, sorry, of glucose, right? So it is a partial breakdown product of glucose. So still energy is present in ethanol, which is not completely harvested yet. That is the reason the amount of energy produced is very less compared to the amount of energy that is produced during aerobic respiration, where each carbon is released in the form of carbon dioxide. So where can you see this kind of process happening? So this kind of process happens in every anaerobically respiring organism. So what all example can we, give for this, right? So if somebody asks you, what is the example for this? You can give the example of yeast, right? So in yeast, what kind of mechanism happens during wine formation, even during bread making and all? What kind of, a process happens? Ethanol fermentation. And what is the byproduct of ethanol fermentation? Carbon dioxide is a byproduct of ethanol fermentation. So that is the first type of fermentation given. Now if somebody asks you, fermentation is an oxidation or reduction process, definitely it is a reduction process, right? Is it a complete breakdown or partial breakdown of glucose? It is a partial breakdown of glucose. What is the reducing agent in fermentation? NADH plus H is the reducing agent in fermentation. Who is the ultimate acceptor of electron in fermentation? Acetaldehyde is the ultimate acceptor of hydrogen or electron in the process of fermentation. How many ATP molecules are produced during fermentation? Only 2 ATP molecules are produced during fermentation. So this is the first type of fermentation, right? So as I was telling you, one more type of fermentation is given in our textbook, that is lactic acid fermentation. So we will just see here itself what is the difference between ethanol fermentation and lactic acid fermentation, right? So we will talk about here lactic acid fermentation. Right? So as the name indicates here, the product will be lactic acid, right? So where all you can see the lactic acid fermentation, this can be seen in the muscle cells, and it can also be seen in the lactic acid bacteria, example lactobacillus, right? So now what happens in lactic acid fermentation? Remember, it is also fermentation. That means it is also an anaerobic respiration. It is also partial breakdown process, right? So let's talk about what are the substrate here, what is the product here. So definitely the substrate here also is sugar, right? What sugar are we discussing here? Glucose, right? So glucose is the substrate here, right? So glucose will be directly converted into lactic acid. See here, there is nothing intermediate here as compared to here that we studied, like acetaldehyde is produced in between, right? So glucose is directly converted into lactic acid. So or else, to be more appropriate, what we will see here, glucose, right, first is converted to pyruvic acid, right? And pyruvic acid is the one which undergo the later fermentation. So this we know. What is the step here? EMP pathway or glycolysis. So here also the first step will be glycolysis, which is common. Then this pyruvic acid will be converted to lactic acid. So there is no reduction in the number of carbon atoms. So that is the reason no carbon dioxide is getting evolved here. That is a very, very important point. The difference between ethanol fermentation and lactic acid fermentation. Carbon dioxide is a byproduct of ethanol fermentation. No carbon dioxide is produced as a result of lactic acid fermentation. So here also it is a reduction process. What is the reducing agent? The same, NADH plus H is the reducing agent, which is converted to NAD plus, right? So it is also a reduction reaction. NADH plus H is a reducing agent, and the product, ultimate product of lactic acid fermentation is only lactic acid. There is no carbon dioxide released in the process, right? And what is the enzyme here for lactic acid fermentation? Lactate dehydrogenase, right? So the enzyme for lactic acid fermentation is lactate dehydrogenase enzyme, right? So this is the two types of fermentation that is listed in our textbook, right? So energetically, when we talk about this lactic acid fermentation in terms of energy released, if we want to know, right, it is also 2 ATP, right? As I told you before, it is only from glycolysis. So in fermentation, the energy that is released during fermentation, that is 2 ATP, it is coming from where? It is coming from glycolysis, because this part doesn't release any energy, right? The reason you can see here, no direct ATP is released, as well as no NADH2 is also released, because NADH plus H, if it is released, it can be converted back to energy, right? But here NADH plus is used, it is not released. So that is the reason when we talk about fermentation, the only source of energy in fermentation is glycolysis or EMP pathway. So in the previous class I told you the net gain of energy in glycolysis is only 2 ATP, right? Always remember, in anaerobic respiration, there is no link reaction, no Krebs cycle, no electron transport system. All the three is only a part of aerobic respiration. When we talk about anaerobic respiration, once the glycolysis is completed, directly the pyruvic acid will undergo the anaerobic process, right? Depending on the organism, different types of product is formed. Like for example, acetic acid can be formed, right? Then we already studied ethanol. There are different types of butyric acid can be formed. All these are formed through the process of fermentation. So this is all about an aerobic respiration process, which is fermentation, which can be seen in different types, which can also be called as fermentation, right? But when we use the term anaerobic respiration, it is a part of the metabolic pathway. So it's like intracellular process. But when we particularly say fermentation, fermentation is done industrially. So it is an extracellular process. So that is the major but both the process right, go hand in hand, right, and both are an aerobic process which doesn't require the use of any energy, right. So that's all for today. Thanks everyone. We'll see in the next class with the next topic.